My name is John Desjarlais. I'm the Chief Scientific Officer at Xencor. Over time, Xencor has emerged as one of the leading Fc engineering companies. So we have a whole suite of different Fc technologies that enhance affinity for various Fc gamma receptors, and we apply those to oncology, autoimmune diseases, and various other diseases. And then more recently, we've developed one of the leading bispecific technologies by engineering Fc heterodimers that enable us to make to put two different selectivities on either side of the Fc to arrive at a novel type of bispecific antibody. Right. Farthest advanced lead is a phase 2 program. It's an anti-CD19 with an enhanced Fc for the inhibitory Fc gamma receptor that's being developed in autoimmune diseases. Morphosis is actually developing. They're now in phase 2 with an antibody, an anti-CD19 with high ADCC that we licensed to them back in 2010. And then we'll have our first 2 bispecifics in the clinic starting next year. So there's a lot of challenges. I mean, I think the new wave is really bispecifics, multi-selectivity, and one of the more important challenges there is being able to manufacture those bispecifics easily, and also just to figure out exactly how to combine the 2 selectivities in the right way with the right affinities to get the properties that you want. And then embedded in that challenge is the fact that there are dozens and dozens of companies trying to all do the same thing. And so I think one of the real big challenges is figuring out what is going to be your niche. What do you have that's novel compared to what any number of pharma companies are also doing. It's kind of what we're doing, and what a lot of other people are doing. It's multi-selectivity. And what's exciting about it is you get to, when you have a bispecific technology, you get to combine 2 targets that don't naturally get coupled in the immune system and see, sometimes crazy things happening. You can make fairly dramatic things happen. For instance, if you recruit a T cell by using a CD3 bispecific coupled with a tumor antigen target, we're starting now to explore also bispecific checkpoint blockade. And we're seeing novel biology emerge when you do those kinds of things. So that's what's exciting is getting to follow brand new biology. I think there's kind of an unmet need in infectious disease that hasn't really been targeted heavily by pharma, and I think there's some emerging results recently that are getting kind of exciting there. And then there's a lot of unmet needs still in oncology, specific types of tumors, tumors that don't really respond to checkpoint blockade, and trying to figure out, how do you actually make something significant happen in those types of tumors. I like to come every year because it gives me an opportunity to see all of the things that are going on in terms of antibody technology development, but also in their application, it allows me to see like a wide swath of what's going on in terms of application to different indications, right? So I see not only the technologies, but also what's going on, kind of some cutting edge stuff going on in terms of different diseases.